Okay, so we'll go ahead and get started. For those of you tuning in perhaps a little later, we are beginning it does say amino acids, but we were supposed to start carbohydrates yesterday. But we will turn to page 751. Some of you have had some exposure to this, but now we will look at this a little further in depth. So carbohydrates on page 751, Gesundheit. Okay. Biochemistry is the study of chemicals and reactions that occur in living things. Biochemical compounds are often large and complex organic molecules. That's why you have a portion called organic chemistry and a separate portion called biochemistry. But their chemistry is similar to that of the smaller organic molecules you studied in chapter 22. Now you will study many important biochemical molecules and learn why they need to stay healthy. And now you will study I'm reading the same thing, and learn why they are needed to stay healthy. Okay. Two of the most common types of molecules that you may know about are carbohydrates and lipids. Okay. These molecules are important parts of the food that you eat and provide most of the energy that your body needs. So carbohydrates is pretty straightforward. That's your sugars. Could be, you look at any type of bottle of soft drink or pop. Perhaps this sounds familiar. You're going to put water in here. You're going to carbonate it, so you're injecting carbon monoxide. Monoxide, hope not. You're going to inject carbon dioxide into here. And then when you do that, if carbon dioxide is placed in here, what happens when you twist the bottle or take an aluminum can and you pry it open? You hear that release of that carbon dioxide gas. So then, yes.
How does like whatever that's called pressure? Yeah, like how do you get that? How do you put that into
the carbon dioxide? Okay, it's in an enclosed container, and then you just inject the gas in here because it's soluble. So then it just dissolves inside the liquid. It will stay that way as long as you have, which leads to our next point. It will stay that way as long as you have something to cover that up. And that might be an advantage on having bottles as opposed to cans, because should you have a can, I don't have one in here, other than the one I used to have, or if I open up a can against people, yeah. And if I open up a can on you, yeah. You don't want me to open up a can. No, probably not. Okay. But anyway, what we're referring to is, why does this have an advantage over an aluminum can? Right, because if you leave this open for an extended amount of time, then you take a drink.
It's flat.
Yeah, because all the carbon dioxide has evaporated out of there. You just might as well just drink sugar water. Okay?
I don't know for sure, but if you shake up a pop can and like pop it open quick, nothing happens.
Oh, well,
Let's try it.
go ahead and try that then. Okay? So carbohydrates, sugar, starches, and cellulose belong to a large group of biochemical molecules called carbohydrates. Are molecules that are composed of carbon, hydrogen, and oxygen, a 1:1 ratio. That's not always the case, but in general, that's pretty common, and provide nutrients to the cells of living things. They are produced by plants through a process called photosynthesis. So sometimes what happens in this class, we will see that different types of, concepts that we have studied will come full circle. And that definitely is true when we talk about proteins in the biochemistry class. Not at this level, but at the next level. Okay? Okay, cellulose provides structure and support for plants, and starch stores energy in plants. Because animals cannot make their own carbohydrates, they must get them from food. Carbohydrates provide nearly all the energy that is available in most plant-derived food. So monosaccharides, that's what we spend the majority of our time on, because we might talk about what a disaccharide is, but we don't draw them. That might be a little above and beyond what we would want to do here. Because one of the things that you see in figure 1 to the right, if you're looking at a molecule of glucose and a molecule of fructose, those are individual sugars, but you can put them together and then you make a whole new category of sugars called disaccharides, which maybe you have heard of one called sucrose. Okay? And in milk, for instance, it's not just glucose. It's two glucose molecules put together, and that we call lactose. Okay? Lactose is a type of disaccharide because it's two different molecules put together. Then if you have maltose, that would be galactose and glucose. So that's something that you would learn next year for those of you who choose to take it. Yes?
Do we have to know those specifically?
No. The glucose, yes. Fructose, yes. But sucrose, maltose, and other disaccharides, no.
How are inorganic materials able to come together to form organic materials?
Magic.
You said inorganic? Because when you say inorganic, we're talking about different types of elements that formed during the Big Bang. So those are all inorganic. But as far as organic molecules, I think a lot of it has to deal with electrochemistry, how different types of energy that's given off between different types of compounds that have nitrogen in it, because that's what an amino acid is, because it has that amine group on it, and that's something that we looked at yesterday. But how do they form organic life forms? That's a little above and beyond than what I could do, other than something kick-started that process where carbon and nitrogen were in the same vicinity. Maybe some sort of catalyst started hooking them together. Then you started making amino acids. Then it's these amino acids that started to make proteins, and these proteins started to form organisms. Okay. A monosaccharide is a simple sugar that is a basic unit of carbohydrate. A single monosaccharide molecule contains 3 to 7 carbon atoms. Okay? Monosaccharide compounds are typically sweet tasting, white solids at room temperature because they have polar hydroxy groups in their molecular structures. Okay? And they are very soluble in water. For anyone who's done that, you can keep adding sugar. Salt is something that's very soluble as well. And the reason that salt is soluble is because you've got chlorine in it. The reason that sugar is in it because it is a type of this polar compound that has all these OH groups on there, which then makes it soluble. Okay? So sometimes I don't know why or how you differentiate between glucose, also called dextrose, and fructose. You got to be a little careful when you're talking about fructose, though, because it's a 6-carbon compound. That's true, but the structure is different. Okay, so then although both of these monosaccharides have the formula C6, and it says H2O and then 6, which is, if you distribute that 6, that gives you your H12 and O6 as well. Okay? Their structural formulas differ then. As figure 1 shows, glucose in water solution forms a ring made of 5 carbon atoms and 1 oxygen atom. Okay? And fructose is a water solution, forms a ring made of 4 carbon atoms and 1 oxygen atom. Notice that both the compounds have 5 OH groups in their structures. Now, that to me really doesn't make any sense when you read it. It makes more sense in this case. Okay? So, if we're talking about these monosaccharides, we could jump right into this here. They're 6-sided figures. And then this other one, okay, fructose. okay, is a 5-sided structure. The reason that we say that these have 6 carbons and 6 carbons here is the devil's in the details, if you want to call it that, because we know that everywhere there's an intersection between lines, what is occurring there? These got these carbon atoms around here. Okay, so then notice there's an intersection here. We don't have a carbon atom there. That's because that is an oxygen atom, just like what we have here. Sorry, what's that? No. Okay, so because if you're talking about alcohol, that just means you have one OH group. This has multiple ones. Okay. So how would we get to something that you see there in the book, and how do you know how to orient all these OH groups? Because some of them are pointing up, some of them are pointing down, okay? What that means is this comes from a specific process or structure we call the Fisher projection formula, okay? And what we mean by that, when it said that there's 4 OH groups in there, these intersections are also carbon atoms. Okay? And then notice that we only have 5 so far. That last one is down here on the end, CH2OH. Because if you look at that figure 1 on page 751, you're going to see in this upper right-hand corner that there's another carbon atom that extends above this, okay? So how do we tie this into this? Well, good question, okay? It's one of the things that you would have to do, and I think when you're gone to your large group contest, it was just 2 of you in here, we could compare this to another monosaccharide. You must have watched that then, okay? Because if we're talking about glucose, okay, it's the orientations of these OH groups, okay? When we say right, left, right, right, we're talking about where these are oriented, okay? And then, yes, you can put these hydrogen atoms in there. It's really not necessary, just like putting this hydrogen atom. That is not necessary. So you might be asking yourself, what type of functional group do we have here? What is a double bonded oxygen with just one hydrogen at the very end of a compound? It's an aldehyde. So we call this an aldose sugar, okay? Because we have this aldose, aldehyde, that's what I'm trying to say. This aldehyde functional unit of this family on the end, because okay, what do you think this 2 lines might mean here? Okay, so when that happens, okay, this sugar is very, very important to process of DNA. Okay, now DNA might not mean anything to you because we say DNA. Okay, we know that this means nucleic, this means acid. Okay. This just means deoxyribonucleic acid. So the reason that's significant is because this is called ribose. So it's this type of structure that's tied into DNA. It's the sugar that makes up portions of DNA. That's as far as we'll go with that because we want to show you that this is a ketose sugar, because have you ever heard of a ketose diet? Well, that means you're eliminating different types of ketones or keto sugars from your diet. And it's maybe not necessarily ribose, but it certainly could be that. But this is, again, as far as we want to go, because this is an aldose sugar, this is a ketose sugar. So then, if that is glucose, some of you had said, well then this is mannose, which it is. But the difference between that is, so we also have an aldose sugar, and then looking at this glucose as opposed to this mannose, there's only one difference between these two structures once we draw them. Okay? So what we see happening occurs right here. So here we said right, left, right, right. This one is left, left, right, right, because of the orientations of those OH groups.
See, and could you do, for the aldose, could it also be left, right, left, left? Is that the same?
Left,
Right,
right, left, left.
like you're just flipping it over.
Well, yeah, it's different types of, we call that an L sugar. That just means that's an enantiomer. Okay? An enantiomer is a mirror image of this. So I don't know if you want to go down that path. Okay. I mean, it's a logical question. It's just something that we discuss next level, what diastomers and enantiomers are that is tied into this. Yes?
Does this connect to those things, those shapes you drew up above?
Yes.
How?
Okay? Because it's the orientations of these OHs that will determine where these are on this compound. Okay?
So that is like that just laid out.
Right. Because how we describe this is just picture this, if this is like a tree, because I made that just a little too big. Okay? And then there. We still have the same thing, right, left, right, right. So what this means then is, notice we have carbon 1, 2, 3, 4, 5, and then 6 all the way down here. Just picture this is like a tree. You cut it off at the trunk, and then it falls over. There's an intermediate step that you would learn next year to where you're taking this information and closing this ring. But we don't do that at this level because it's just a little too complex. Because what you do, okay, someone had said something about Greek letters.
Alpha.
Okay, that's one of them. And then
Beta.
beta is the other. So what I want you to do is pick one of those symbols.
Alpha goes up, beta goes down.
The other way around.
What the?
Okay? Because alpha, this here, means that this OH group is going to be down. Okay? And chances are that's how they have this wrote out too. Okay? So then this is carbon number 1, just like this is carbon number 1.
Hold on, I thought you said that's aldose though. Is that something different or is that glucose?
This here?
Yeah.
Glucose is an aldose sugar. It's just like saying that if you have an artidactylian, okay, which means an even-toed, even-hooved mammal, that you could say that, okay, deer and cattle, they both have 2 hooves, but they're not in the same family.
So aldose is like the broader.
Aldose is the very broad. Both of these are aldose sugars. We could say aldose for this one too, but it's just it's just known as mannose because of how this is located there. Okay. So then here's our second, third, fourth, fifth. Because this is on the right, where is that OH group located on carbon number 2?
On the top.
Where is it located on the second carbon? Because if it's on the right, that means it goes down. So what clue did we give in the video for those of you who were here? We said 2 ways you could memorize this. RD. You can say either Reader's Digest or Research and Development, R and D. Because if it's on the right, the reader part, the D or digest means that it's down. So R and D. And then if it's not on the right-hand side, it's on the left, it can't be down because then it would be over here. So then if it's not down, it must be. So where do you see that in your book on page 751? Where's that OH group? Is it down or is it up?
Down. Up.
It's down?
Oh, I'm looking
Okay.
at
Yes, it is up. Okay. Well, it could be a misprint too. So then look at carbon number 4. It's on the right. So is it up top here or is it down? It's down below. Now, this is the tricky part. Again, there's a couple intermediate steps that you would say, well, that OH group is down here. Well, it was at one time. You could ask any of the seniors, okay? There's a couple. Say that again.
The OH has to be on the outside
Of the ring? Yes, outside.
So once you get to the top, you flip it over.
In a matter of speaking, again, there's a couple intermediate steps that we just don't cover at this level. Okay? Because at one time, this was an OH group here. Okay? Because remember, this was double bonded. Okay? Because where's that double bond now? Well, that's why we say in this process of reduction, because that's what happens. You're in reduction, you're gaining electrons, and that gain of electrons came from this OH group, okay? So this O is this one right here, okay? This hydrogen ended up coming down here, and that's how that ends up as OH, because this hydrogen is still here. It just hasn't gone anywhere. Okay? That's what I mean. There's some intermediate steps that's a little more complex than what we want to do at this level. Okay, so the main takeaway from this is taking this information, if it's on the right, it's down. If it's on the left, it's up. And when you get to this point on carbon number 5, yes, it was down at one time, but this oxygen is here now. So where is carbon number 6 in accordance with your figure?
Up there.
Up top here, so CH2OH. That would be carbon number 6. Okay, I have something brewing in the lab. I'm trying to perfect the lab that we worked on, so I need to take just a short break. Okay, so one of the things we had done with your classmates, I had to go next door because we're trying to perfect an activity that we're doing for the elementary students tomorrow, and I think it got too hot and I left it in there for too long because we're just trying to multitask, but it'll be okay. All right, so we left off with talking about where carbon number 6 is located, which is up here. Okay? Don't worry about this. We're just using that as an example. That you have ketose sugars and you have aldose sugars. And that's what we see here. This is an aldose sugar. This is an aldose sugar because that double bonded oxygen is on the first carbon. So what we would look at and interpret then, okay, we'll leave both of these up here. Okay, so we'll go ahead and put all these in here. And again, 1, 2, 3, 4, 5, and 6. For certain, we know that CH2OH is here. So now you need to pick a letter. Okay, so then if alpha was down, well then beta is up here. Okay, so then think of your clue, either Reader's Digest or, yes.
Where did we get that first one from?
This one here?
Yeah.
Okay. I'll try to keep this short. as possible.
Will we have to know that for the test?
What's that?
Will we have to pick one of those and then do it?
You would have to, if it says beta D mannose, you would have to put that up there. Okay? So beta is up, alpha is down. Okay, so a question was, how do we know where carbon number 1 is? Okay, well, there's carbon number 1 because it's an aldose sugar, right? If it's bonded on the end, that means there's a double bonded oxygen with a hydrogen. Double bonded oxygen with a hydrogen. Then we can just go right around the horn here. Okay. Oops, that's not correct. Now, do we really want to know this?
Do we need to?
I want to just know like what sugar.
Okay, because if this is carbon number 1, just like what we have here, okay, then there's some similarities in here. This is on the left side, so where's this OH group located then? It's located up. Okay. What about this one? That's also up. Okay. And then what about this one? Okay. And then see what happens is we see some similarities here. This one was up, this one was up, this one was down, this one was down. Okay. Because here we see it was on the left and then the right and the right. Left, the right, and again, this was down here at one time. Okay? And what happens is here's carbon number 6, way out here on the end. Okay? Here's carbon number 6. Because what happens, and I really can't explain it other than this OH group wants to get closer to this carbonyl carbon, because yesterday, remember, we talked about carbonyl atoms. Those occur in aldose or aldehydes, ketones, esters, okay, acids. What do all those have in common? They've got a double bonded something. They've got a double bonded oxygen. So what happens in this intermediate step is this ends up over there. Okay, so this wants to be closer to the carbonyl group. I suppose there's some electronegativity reasons for that when all these different types of substances are getting together would be my guess. Okay, then now this looks as it should. Then what happens is, remember we talked about reduction, or what? This gets reduced or electrons are gained. This, we had a box around that. This is all by itself now because this hydrogen is coming down here and it's breaking that double bond. So then what we see happening then is this is why this is an OH group now. It's because that hydrogen broke that double bond. So now you can see these side by side. So I don't know if that helped, but that is the real reason why you see this as C double bonded oxygen with a hydrogen, and why this really was here at one time, but it ended up at the end because they flip-flop around.
So how come there's 3 OH on the top when 2 OH on the left?
Okay, so 3 OH on this one, and this one.
No, the one to the very right.
This one?
Yeah.
Okay.
1, 2.
Well, again, that used to be this, okay?
Oh,
Because what happened is this hydrogen came down here and it broke that double bond to form an OH group here.
so understood.
So now that hydrogen is attached to this because it was a reduction reaction. And then that hydrogen, it never left, it still just stays there. So now when you look at these side by side, they're almost superimposable except for here, okay, and at the second carbon. And we know that because the second one was on the right, here the second one was on the left. The third one was on the left, the third one's on the left, the fourth one's on the right, The fourth one's on the right. And of course, that really doesn't matter. You could almost pick this up, stick it on top of here, and you'd almost have the same molecule except for that location. You could even say this is alpha D mannose. Then it's almost identical. Then that's the only spot that is different. Who's lost? Front row, are we on the bus?
I'm just kidding, I'm on the bus.
Okay. Middle row,
I'm just kidding, I actually kind of.
back row. Okay. I think maybe that was enough.
So you have 4 rows, but you really only placed 3 of them.
Right.
No, 'cause what?
That's right. Because this one, remember, used to be here. Then it moves over to here, and then that goes through a process of reduction where that hydrogen ends up down there.
So really the first one.
The fourth one, only the second, third, and fourth ones, second, third, and fourth, we know that we can for sure take from this.
So the 5th one is just from the very bottom one.
This one? What about it?
It takes the one at the very bottom, the CH2OH.
No.
Well, it puts that on top.
It replaces that up here. Yes. Yep. Okay. We didn't quite get to fructose yet, but that's okay.
That's all right.
So now, tomorrow it still says review, which we'll review this process. Then Friday we'll talk about.
All of it.
No, this I'm a day ahead of ourselves. Tomorrow we'll go over this. Thursday, okay, we'll start talking about amino acids. Thursday and Friday we'll start talking about amino acids rather than saying, oh my gosh, we're going to have a test over that this week. Okay, it's a fairly big one, yes, but it'll be some of your last ones that we take.
That's enough.
Okay, so that's all I have. We'll regroup and catch up to you next time.